Digestion of food. The process of digestion is accomplished by mechanical and chemical processes. The buccal cavity performs two major functions: mastication of food and facilitation of swallowing. The teeth and the tongue, with the help of saliva, masticate and mix up the food thoroughly. Mucus in saliva helps in lubricating and adhering the masticated food particles into a bolus. The bolus is then conveyed into the pharynx and then into the esophagus by swallowing or deglutition. The bolus further passes down through the esophagus by successive waves of muscular contractions called peristalsis. The gastroesophageal sphincter controls the passage of food into the stomach. The saliva secreted into the oral cavity contains electrolytes Na plus K plus Cl minus HCO3 minus and enzymes. Salivary amylase and lysozyme. The chemical process of digestion is initiated in the oral cavity by the hydrolytic action of the carbohydrate splitting enzyme. The salivary amylase, about 30% of starch, is hydrolyzed here by the enzyme. Optimum pH 6.8 into a disaccharide maltose lysozyme present in saliva acts as an antibacterial agent that prevents infections. The mucosa of stomach has gastric glands. Gastric glands have three major types of cells, namely mucous neck cells which secrete mucus, peptic or chief cells which secrete the pro-enzyme pepsinogen, and parietal or oxyntic cells which secrete HCl and intrinsic factor essential for absorption of vitamin B12. The stomach stores the food for 4 to 5 hours. The food mixes thoroughly with the acidic gastric juice of the stomach by the churning movements of its muscular wall and is called the chyme. The proenzyme pepsinogen, on exposure to hydrochloric acid, gets converted into the active enzyme pepsin, the proteolytic enzyme of the stomach. Pepsin converts proteins into proteoses and peptides. The mucus and bicarbonates present in the gastric juice play an important role in lubrication and protection of the mucosal epithelium from excoriation by the highly concentrated hydrochloric acid. HCl provides the acidic pH 1.8, optimal for pepsins. Renin is a proteolytic enzyme found in gastric juice of infants which helps in the digestion of milk proteins. Small amounts of lipases are also secreted by gastric glands. Various types of movements are generated by the muscularis layer of the small intestine. These movements help in a thorough mixing up of the food with various secretions in the intestine and thereby facilitate digestion. The bile, pancreatic juice and the intestinal juice are the secretions released into the small intestine. Pancreatic juice and bile are released through the hepatopancreatic duct. The pancreatic juice contains inactive enzymes trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, lipases and nucleases. Trypsinogen is activated by an enzyme enterokinase secreted by the intestinal mucosa into active trypsin, which in turn activates the other enzymes in the pancreatic juice. The bile released into the duodenum contains bile pigments, bilirubin and biliverdin, bile salts, cholesterol and phospholipids, but no enzymes. Bile helps in emulsification of fats, that is, breaking down of the fats into very small micelles. Bile also activates lipases. The intestinal mucosal epithelium has goblet cells which secrete mucus. The secretions of the breast border cells of the mucosa along with the secretions of the goblet cells constitute the intestinal juice or succus entericus. This juice contains a variety of enzymes like disaccharides, dipeptidases, lipases, nucleosidases, etc. The mucus along with the bicarbonates from the pancreas protects the intestinal mucosa from acid as well as provide an alkaline medium pH 7.8 for enzymatic activities. Some mucosal glands, Brunner's glands, also help in this. Proteins, proteases and peptones, partially hydrolyzed proteins in the chyme, reaching the intestine are acted upon by the proteolytic enzymes of pancreatic juice as given below. Proteins, peptones, proteases are converted into dipeptides in the presence of trypsin, chymotrypsin and carboxypeptidase. Carbohydrates in the chyme are hydrolyzed by pancreatic amylase into disaccharides. Polysaccharides that means starch are converted into disaccharides in the presence of amylase. Fats are broken down by lipases with the help of bite into di and monoglycerides. Fats are converted into diglycerides in the presence of lipases and are converted into monoglycerides. Nucleases in the pancreatic juice acts on nucleic acids to form nucleotides and nucleosides. Nucleic acids are converted into nucleotides in the presence of nucleases and then converted into nucleosides. The enzymes in the succus entericus act on the end products of the above reactions to form the respective simple absorbable forms. These final steps in digestion occur very close to the mucosal epithelial cells of the intestine. Dipeptides are converted into amino acids in the presence of dipeptidases. Maltose is converted into glucose plus glucose in the presence of maltase. Lactose is converted into glucose plus galactose in the presence of lactase. Sucrose is converted into glucose plus fructose in the presence of sucrase. Nucleotides are converted into nucleosides in the presence of nucleotidases and then converted into sugars plus bases in the presence of nucleosidases. Di and monoglycerides are converted into fatty acids plus glycerol in the presence of lipases. The breakdown of biomacromolecules mentioned above occurs in the duodenum region of the small intestine. The simple substances thus formed are absorbed in the jejunum and ileum regions of the small intestine. The undigested and unabsorbed substances are passed on to the large intestine. No significant digestive activity occurs in the large intestine. The functions of large intestine are absorption of some water, minerals and certain drugs, secretion of mucus which helps in adhering the waste, undigested particles together and lubricating it for an easy passage. The undigested and unabsorbed substances called feces enters into the cecum of the large intestine through ileocecal valve, which prevents the backflow of the fecal matter. It is temporarily stored in the rectum till defecation. The activities of the gastrointestinal tract are under neural and hormonal control for proper coordination of different parts. The sight, smell and or the presence of food in the oral cavity can stimulate the secretion of saliva. Gastric and intestinal secretions are also similarly stimulated by neural signals. The muscular activities of different parts of the alimentary canal can also be moderated by neural mechanisms, both local and through CNS. Hormonal control of the secretion of digestive juices is carried out by local hormones produced by the gastric and intestinal mucosa. Calorific value or protein, carbohydrate and fat. The energy requirements of animals and the energy content of food are expressed in terms of measure of heat energy because heat is the ultimate form of all energies. This is often measured to as calories Cal or joule J, which is the amount of heat energy required to raise the temperature of 1 gram of water by 1 degree centigrade. Since this value is tiny amount of energy, physiologists commonly use kilocalorie kcal or kilojoule kj. 1 kilocalorie is the amount of energy required to raise at the temperature of 1 kg of water by 1 degree centigrade. Nutritionists traditionally refer to kcal as the calorie or joule, always capitalized. The amount of heat liberated from complete combustion of 1 gram food in a bomb calorimeter, a closed metal chamber filled with O2. Is its gross calorific or gross energy value. The actual amount of energy combustion of 1 gram of food is the physiologic value of food. Gross calorific values of carbohydrates, proteins and fats are 4.1 kilocalorie per gram, 5.65 kilocalorie per gram and 9.45 kilocalorie per gram respectively, whereas their physiological values are 4.0 kilocalorie per gram, 4.0 kilocalorie per gram and 9.0 kilocalorie per gram respectively. Absorption of digested products. Absorption is the process by which the end products of digestion pass through the intestinal mucosa into the blood or lymph. It is carried out by passive, active or facilitated transport mechanisms. Small amounts of monosaccharides like glucose, amino acids and some electrolytes like chloride ions are generally absorbed by simple diffusion. The passage of these substances into the blood depends upon the concentration gradients. However, some substances like glucose and amino acids are absorbed with the help of carrier proteins. This mechanism is called the facilitated transport. Transport of water depends upon the osmotic gradient. Active transport occurs against the concentration gradient and hence requires energy. Various nutrients like amino acids, monosaccharides like glucose, electrolytes like Na+ are absorbed into the blood by this mechanism. Fatty acids and glycerol being insoluble cannot be absorbed into the blood. They are first incorporated into the small droplets called micelles which move into the intestinal mucosa. They are reformed into very small protein coated fat globules called the chylomicrons, which are transported into the lymph vessels lacteals in the villi. These lymph vessels ultimately release the absorbed substances into the bloodstream. Absorption of substances takes place in different parts of the alimentary canal like mouth, stomach, small intestine and large intestine. However, maximum product absorption occurs in the small intestine. A summary of absorption, sites of absorption and substances absorbed is given in table 16.1.